Hi, my name is Jaleel Sanjak, and I'm a graduate student in evolutionary biology at the University of California, Irvine. Did you know that we are all mutants? Humans are 99.9% identical, yet we are all as unique as snowflakes. It's cute, right? Inside each of us is a personal genetic codebook called a genome. This genome codebook is so big, 3 billion characters, that 1/10 of 1% difference amounts to 3 million differences just between you and I. The main book chapters are called genes, and they describe the main tools used to build a human. Mutations are kind of like typos in these instructions, in that they can change or destroy the meaning of words. We also call mutations genetic variants, in that they are places where people's genomes vary. Some genetic variants are unique to a single individual, while others are shared in large groups of people due to common ancestry. This has a major public health implication. Many common diseases, such as diabetes, run in families. Thus, there are genetic variants that partially determine differences in disease risk. We may be able to use our understanding of genetic variation to learn about diseases. We may in turn use that to help predict patient outcomes and provide better treatments. For the past decade, researchers have tried to uncover the genetic variants that determine disease risk. Despite some success, this goal has not been achieved. But why is that? I try to help answer that question using the theory of population genetics. This is the mathematical synthesis of Gregor Mendel and Charles Darwin. It is how we relate molecular genetics to large-scale evolution. Mutations arise at random and are passed on according to Mendel's rules. Over time, genetic variation accumulates in the population, potentially leading to the origin of new species. Importantly, this theory also predicts how the genetic variation within a population might be structured. By structure, I basically mean who shares which mutations. Can predictions from population genetics show us how to uncover the genetic basis of human disease? Statistics, the ugly troll under the bridge of science, rears its head here. Finding a genetic variant that contributes to disease risk starts with a statistical correlation. We take two groups of people, one that has diabetes and one that doesn't. Somehow, we compare the genomes of these two groups of people, and we count up the genetic variants to see which is more prevalent in one group than the other. It sounds easy, right? But we ought to slow down and think for a second. We know that everyone is a special snowflake. At least my mom says I am. So, some genetic variants will differ between the two groups just by chance. Going back to the codebook example, imagine that somebody wrote a random side note in the margin of the book, say, information from a telemarketer call or something like that. That note would appear as a difference between two books, but it's not actually related to the instructions. People's genotypes at genetic variants that are physically close to each other in the genome are highly correlated. And so it's hard to know which one matters. Also, due to common ancestry, a sample of genomes can never be truly viewed as random. And today, due to financial limitations, we are faced with a choice. Read a few codebooks thoroughly or skim a much larger number. We don't know which would be better. So we don't know which data to collect, and we don't know how to analyze it. But we do know that the answer to those questions depends heavily on how the underlying genetic variation is structured. Thus, it depends on population genetics. The theory can tell us what to expect and what to be surprised about from experiments. But real experiments cost time and money. So we put the theory to work by performing experiments on a computer. We do this by assuming a model for how mutations affect the disease and how the disease affects evolutionary fitness. For example, imagine that our ability to produce insulin is very important and finely tuned. This would suggest that mutations that either increase or decrease insulin production would generally be bad. The worse the mutations are, the less frequent they will be in the population. The more things that actually contribute to insulin production, the less any one mutation is likely to matter. Using the theory and a supercomputer, I can make those ideas explicit. We simulate the evolution of large populations where disease risk is due to certain mutations. I can then perform experiments on those simulated populations. I can ask, how would we know which mutations actually cause disease? How is the genetic variation structured overall? We can compare outcomes using different statistical and experimental approaches. We find that if diseases are even a tiny bit bad for you evolutionarily, that skimming the codebook is unlikely to work. If diseases are bad, the mutations that cause them will be rare. And if the mutations are rare, then there must also be many different types. Thus, you need to read each codebook thoroughly to find all the mistakes. Also, statistical tests that use information from many genetic variants simultaneously work the best. This is because it is unlikely that the same genetic variant is responsible in all cases. Many different typos can make a sentence illegible. An interesting new approach to looking at genomes is currently in use, called exome sequencing. This allows us to thoroughly read just some key sections, the genes. This is kind of like reading the Cliff Notes version of the genome codebook. Moving forward, I will tweak my simulations for application to these data. In doing so, we will continue to gain fundamental insight into the evolution of genetic disease. Perhaps some of you have heard of precision or personalized medicine. A big part of that is tailoring medical advice to you based on your genome. For example, you may possess a mutation that makes alcohol more harmful to your brain than it is to the average person, and choose to lay off the booze. This is very likely to be part of our future. But first, we need to understand which differences in our genomes result in real differences between us. I think we can achieve this goal, but we have to let the evolutionary process be our guide. For now, long live the mutants!